Hello, everyone. I am at the school in the lobby, so hopefully this will work and you can hear me. Um, we're going to go over macromolecules today. That's the activity we did in class on Wednesday. So let us start by with our Bible verse this morning. So our Bible verse is here. Our Bible verse for today is, "For his invisible attributes, that is, his internal power and divine nature, have been clearly seen since the creation of the world, being understood through what he has made. As a result, people are without excuse." Romans 1:20. Okay, so today we're going to talk about new share. Here we go. Macromolecules. The first one that we'll talk about are the carbohydrates. So in the lab activity that we did, one group had a tray of glucose beads, a tray of galactose beads, and a tray of fructose beads. And then they had white, red, green pipe cleaners and some small white pipe cleaners. And this is what they made. They made disaccharides and polysaccharides. So let's look through the questions here. And I've uploaded these sheets onto the materials on Jupyter Ed. So you can print these out at home and make notes as needed. So the first thing that we need to realize is that carbohydrates are made up of monomers called monosaccharides, and those are the simple sugar molecules. And we have three of those in our activity. We have glucose, galactose, and fructose. So if you want to know what is the difference between the three groups, well, the glucose beads were white, the fructose beads were clear, and the galactose beads were pink. And so the color was different, but the shape of the beads were the same. So it gives you down here an excellent chart of what the monosaccharides look like. This one is your glucose molecule, the molecular structure. You see that there are 1, 2, 3, 4, 5 carbons, and then you have an OH group here, an OH group here, you have another carbon here, so 6 carbons total, and you have oxygen atoms and hydrogen atoms that make up carbohydrates. In this one, you have 6 carbons, 6 oxygens, and 12 hydrogens. And that makes its molecular formula C6H12O6. Now, if you look between the glucose and the galactose, there is a difference. It's right here with the shape, the way that the structure. And the thing that you need to remember in science is that structure dictates function. If you change the structure of a molecule, really if you change the structure of anything, it changes the function of that structure. So you see here that galactose looks different from glucose, but it still has 6 carbons, 6 oxygens, 12 hydrogens, and the molecular formula is C6H12O6. Then if we look at fructose, we see some major differences with fructose in the structure. However, it's still 6 carbons, 6 oxygens, and 12 hydrogens, so it's still C6H12O6. Now, we call all of these isomers because they're the same number of molecules, the same molecular formula. So they have the same number of molecules, the same type, but the structure is different. So that's called an isomer, and that's a vocabulary word. The bonds between carbon atoms and other carbon atoms store a lot of energy. When broken, they release a lot of energy. How many carbon-carbon bonds are in each monosaccharide? There are 5 carbon-to-carbon bonds. If you look at the structure, you see that there's one that's bonded to oxygen here. So there are 5 carbon-to-carbon. 1, 2, 3, 4, 5. 5 carbon-to-carbon bonds. Those can be broken and release a lot of energy. Now, monosaccharides join together to make a disaccharide. We talked about how the prefix di means two. And so we have monomers, 2 monosaccharides make a disaccharide. And what is the reaction that takes place when 2 small molecules lose the water and form a larger molecule? Well, if you lose water in your body, we say that you're dehydrated. And if you lose water in a reaction, we say that it is dehydration synthesis. And so if you look at this reaction, it would be a monosaccharide plus a monosaccharide joined together, bond together to make a disaccharide plus a water. A water is released or produced. And so here we have these pipe cleaners. And if we take a glucose and a fructose on this little piece here, then this is the disaccharide sucrose. So this made sucrose. If we take galactose and glucose and bond those together, releasing the water in dehydration synthesis, we bond these 2 together, galactose and glucose, we form lactose. Then if we look on down and we have glucose and glucose, then we form maltose. So there's maltose. So now we have 3 disaccharides, maltose, lactose, and sucrose. Okay, these are disaccharides. So if we have a white and white, it's maltose, a white and a pink on ours, it was lactose, and a white and clear sucrose. So now we can join monosaccharides and form a very long chain of monomers, and we can make carbohydrates. So these are very long chains of monomers, monosaccharides, to form a polymer called a polysaccharide. Okay, poly means many, saccharide means sugar, because it's made up of many simple sugars. Cells connect in many monosaccharides together to store them and to save them for later use. Some cells also string together monosaccharides to form building materials for their cell walls. Different polysaccharides are formed when monosaccharides are combined in different combinations or structures. So let's look at 3 different types of polysaccharides. The first one is cellulose. So we took a white pipe cleaner and we put all of the glucose monomers onto this pipe cleaner, white pipe cleaner, to make a cellulose. Cellulose is indigestible for most humans, for most animals, including humans, but there are animals like cows that can chew cud and digest cellulose. Okay. This one on a long white pipe cleaner, let's see. Sorry, cellulose was on the green pipe cleaner. So here's cellulose on a green pipe cleaner. Here is starch on a white pipe cleaner. And this is, animals can break this down. This is what we eat, starch. We break this down for energy, to use for energy. In everything, corn, any corn product that's used, corn to make it, potatoes, beans, rice, peas, all have starch. And we break this macromolecule down for energy. Then we have glycogen. Now, glycogen, let's see if I can make glycogen for you. Maybe I can. Okay, so we have fill a long red pipe cleaner with white beads. So I've got a pipe cleaner, red pipe cleaner filled, and then connect the end of one pipe cleaner to the middle and create a branch. Okay, so I'm going to create a branch here. Oh, I just dropped beads all over the floor here. Okay, there we go. And then if I make this kind of fold back up through here, I can make a glycogen molecule for you. Okay. So here is a glycogen molecule. Okay. So we have repeating units of monomers. These are all glucose, but in the glycogen, we have a branch on our chain, on our polymer chain. Okay, and this is how animals store glycogen. If we've used all of the energy that we need from what we've eaten, then we can store this in our muscles for later use. And we can put it, we can store it in the liver. Okay, so to recap on carbohydrates, we have carbohydrates are made of monomers called monosaccharides. This monosaccharide is called glucose. Glucose can go together in long chains called polysaccharides, which is a long chain of many sugars. So this one is starch on a white pipe cleaner. This long chain is cellulose in plants for cell wall, cell structure. And then this one is glycogen, which is what the body uses to store energy that we don't need right away. So those are your 3 carbohydrates. Okay. Now I'd like for you to read over this last page about the rabbits and hydrolysis, which is basically the reaction that we use to break down the polysaccharide. So we use hydrolysis to break this down, to use the energy from the bonds. And don't forget that we have different kinds of disaccharides that we can make when we join 2 monomers together. We can make a disaccharide. And all of these are sugars that combine to make polymers such as starch, glycogen, and cellulose. Okay? Okay, the next one I would like to show you. Let's go to the proteins. Proteins, here we go. So in this one, you also have this one on your materials. So these are the proteins. So what we had a bunch of different beads in our class. One group had a bunch of different beads, and they were building proteins. Okay. So, okay. Oh, it's fine. It's okay. Please do. Please do. You're fine. It's okay. All right. So amino acids. These beads. Is my car in the wrong place? Oh my word.
Emily, you look like you're in for seniors that are leaving.
I do have a senior pass.
Okay.
I do. Well, I have a junior pass.
Yeah, seniors. Some of the seniors have special parking because they don't stay all day.
I'll move.
It's not urgent.
I'm zooming, but I'll
That's fine. Whenever. It's not urgent.
Okay, I always put my car in the wrong place. So these beads represent amino acids, the monomers of the polypeptides and proteins. So how are your beads different? Well, they're all different. They're different sizes, different shapes. Some have variety in different colors. So these beads are very different from one another. We don't have, it's not like the carbohydrate that we had here where all the beads look the same and we're repeating units of the same monomer. These are all completely different. So this is what proteins look like. All the proteins do have a hole that goes through the center of the bead. So they are similar in some way. And if we look at this, each amino acid is similar in their basic structure. So you have an amine group, a hydrogen, a carboxylic acid group, and an R group attached to your central carbon. What does each amino acid have in common? There you go. We just said it. These are the things that the structure, basic structure is what they have in common. And the thing that makes them different is this R group right here. And the amino acids got their name because it's from the amine group. And this is an acid, so amino acid. Bonds between carbon atoms and other carbon atoms store a lot of energy. How many carbon-carbon bonds are in each amino acid? You only have one, and it's right here. So that's your only carbon-carbon bond, meaning that these contain less energy than our polymers, because in our monosaccharides, our polysaccharides, because these had lots and lots of carbon-carbon bonds. So your body would want to use the energy from these monosaccharides on this polysaccharide before using energy from your protein. So two amino acids come together and form a dipeptide. Same way, it's going to lose a water. So if you have an amino acid plus an amino acid, you're going to get a dipeptide, meaning two amino acids together, and you're going to get water. Same reaction that we saw in the carbohydrates. The bond between the two amino acids in the chain is called a peptide bond. So a polymer chain of amino acids strung together with covalent bonds is called a polypeptide. So here is a polypeptide, okay? And you see here in this reaction, there's your H2O. Your H2O is removed, and here's the new bond that replaces that H2O. There it is right there between the carbon and the nitrogen. So you have one amino acid here, and you have another amino acid here. If you lost another water, you could connect another amino acid and make a very long chain, a polypeptide. Okay, so I could go ahead and name, I have a brown round, a cream round, a pink round, a blue round, a green round, a yellow round. Then I have a larger round natural wood. So this is the primary structure. Whatever sequence your amino acids are in dictates the function. Remember, we said structure dictates function. So if I move these two amino acids back here, does that change the function of this protein? Yes, it does, because I changed the structure of this protein. I changed the structure of it, so now the function of this is different than it was before. Polypeptides are produced from blueprint instructions stored in the DNA cell, in the cell's DNA. Each type of polypeptide is different, just as different machines build different instructions for different instructions are different. The structure and function of a polypeptide is determined by the sequence. That's what we just said. And if I had another one, we could go through all of the structures, but basically if we have it like this and then we twist it, we get the secondary structure, and then we can get the tertiary structure. Here it is all, if I had another one, we could make it more like the tertiary, and then finally you get to the final structure with multi-peptide polypeptide proteins. So cells often use proteins to do jobs around the inside and surface of the cell, and each protein has a specific function. Sometimes proteins only need one polypeptide chain to function. Often proteins need more than one polypeptide chain to function. The two polypeptide chains you folded together now form a functional protein with two polypeptide subunits. Okay. All right. And now I would like for you to answer these questions. Just read over them. This last page of the protein packet that I put onto the material. So far we've studied the protein, we've studied the carbohydrate. So let's do fats. Let's do lipids next. I've got to plug in my computer before it dies. It's always something. And then I have to move my car, but it's okay. I've moved my car 3 times this week, last week and this week. I never park in the right place. I need a tutorial on where to park. Okay. Are you serious? There's no cord outlet here. Oh, yeah, there's one. Okay, we're gonna move down here to the other side of the table. We're charging. Okay, it's a good thing that I like to laugh because this is kind of funny. Okay, so now we will move on to our next. And I said we would do lipids next. So we're doing, this is our 3rd out of 4 macromolecules. So let me share this with you. Okay, lipids. So with the lipids, these groups had 5 beads. They had some half pipe cleaners. They had some small pieces and some medium pieces. And so let's read through what this is what this group made. Okay, so here's one example of a lipid. Okay, so let's read through this. So your medium length pipe cleaners represent fatty acids and your long chains of carbon and hydrogen molecules. So these are your fatty acids, which are long chains of carbon and hydrogen molecules. And then you notice that you have different colors in the 2 groups of medium-sized pipe cleaners. Well, let's go on down here. So that, okay. Oh, yes. We had some that were straight and we had some that were bent. So this is the difference between these pipe cleaners. We had some that were straight like this, straight as it can be with pipe cleaners. Then we had some that were bent. Okay. Each fatty acid has a unique structure. Let's look at those. So the molecular structure of a saturated chain of a fatty acid has no double bonds. Everything is single bond to another carbon. So this makes this fatty acid have 9 carbons, 2 oxygens, and 18 hydrogens. So C9H2O18. However, this unsaturated, which is bent, so this is our unsaturated fatty acid, it has 9 carbons, 2 oxygens, and 16 hydrogens, because if you look here, 2 hydrogens have been removed and another bond has been placed between the carbons. It's a double bond. So you see the we had 18 hydrogens here, oxygens, wait, we had 2 hydrogens here, 18 oxygens here. We've removed it here. Okay, this is not right. This key is not right. This should be C9H18O2. And this should be C9H16O2. Wow, that threw me for a loop. Yes, this is wrong. So change that on yours. This should be C9H18O2, and this one's lost 2 hydrogens, so it would be C9H16O2. So in other everyday application, what does the word saturated mean? Saturated means it's full of something. So in this case, it is full of hydrogens. There's nowhere else for a hydrogen to go. Saturated fatty acids are fatty acids that are full of hydrogens and have no double bonds. What is the difference between the 2 molecules? Well, the one on the left has hydrogens on different sides of the double bond, and one on the right has hydrogens on the same. So if you look here, one we call trans, and then this one we call cis. So these are definitely isomers of the unsaturated fatty acid. The bond between carbon atoms and other carbon atoms store a lot of energy. When broken, they release a lot of energy. So each fatty acid in the picture has 8 carbon-carbon bonds. So it can release that much energy. Some fatty acids have 30 or more carbons in a chain. And if there were 30 carbons, then this is what we would have, C30H60O2. That one's correct. That's correct. Okay. So let's look at the monosaccharide and the fatty acid, amino acid and fatty acid combo below. So we have the monosaccharide, which we should recall from our carbohydrates over here. So here is there's a monosaccharide, and it has 5 carbon-carbon bonds. It's a carbohydrate. These form together to form this, which is a carbohydrate. So these are bonds that are very easy to break. The amino acid only has 1 carbon-carbon bond. It forms proteins. This was our protein. Only 1 carbon-carbon bond in our protein, and it is very difficult to break that carbon-carbon bond. And then here's our fatty acid with 9 carbon-carbon bonds. It is a lipid or a fat, and it is difficult to break these bonds. So which molecule would the cell use up first for quick energy? First, you're going to use your sugars, your starches, your carbohydrates. Then you'll use your fatty acids next. And then finally, your body will start using the amino acids, your proteins. Okay, and that here it explains it. It's because of the number of carbon-carbon bonds. So we're going to find our short green pipe cleaner, and that's going to represent glycerol. So we have our short green glycerol here. And a triglyceride is made up of 3 fatty acids connected to a glycerol. So we have 3 fatty acids connected to a glycerol molecule, which is right here. And to build a triglyceride, again, what are we going to take out? We're going to take out water. Only this time we're going to take out 3 waters. So you have your glycerol here, and then your 3 fatty acids. So this forms a triglyceride, and then 3 plus 3 water molecules. Okay, so there's your triglyceride. They are the main components of oils and fats. Saturated fatty acids have more hydrogen and are more likely to be a solid at room temperature. These are things like butter, lard. Those are things that are solids at room temperature. Oils are your unsaturated fats usually, and those are usually liquids at room temperature. And then do these things dissolve in water? You know, we talked a lot about water in class and about how it looks like Mickey Mouse, and it has a positive side and a negative side, and it will dissolve anything that has a charge. Well, will water dissolve oil? No. Will it dissolve fat, butter, no, because those are nonpolar. They do not have charges. They do not have charged ends based on their structure. Now let's talk about a phospholipid. A phospholipid is made of two fatty acids and a phosphate group. So let's make one of those. So I'm going to take off one of my fatty acids, and I'm going to get a, let's see if I have one. Get a little pink bead here. So this represents my phosphate group. So I'm going to put that there and twist it to the side like this. So now I have a phospholipid. I put a phosphate group on it, and then I still have two fatty acids. Okay, fatty acid chains, when by themselves, do not dissolve in water. They are hydrophobic. They have a phobia of water. They do not dissolve in water. However, the phosphate group is hydrophilic. It loves water. So this is one of the things that God designed to help us in many areas, because it has a head is the phosphate group, and the two fatty acids are the tail. And so this part right here is, oh, you're fine, you're fine. I had to come over here for the cord. So, and I left all this here at school, so I couldn't do my thing until today. So this one is your phosphate group. It's hydrophilic, and these are hydrophobic, your fatty acids. So look what happens. It's so cool. This is how we make our cell membrane. So this is one side would be like this, and the other side would come up to it like this, and your hydrophobic ends would come together, and then your phosphate group, your hydrophilic ends, would be on the outside. And this is how we make a cell wall, or a barrier that separates the outside from the inside. So that's pretty cool. And here's another lipid, cholesterol. It forms a ring. You can see it there of carbon. Every bend on this structure is a carbon. So everywhere there's a bend, there's a carbon atom. And so this would be a very complicated shape to make with pipe cleaners. So there's not additional reading for this particular packet. So let's move on to our last macromolecule of the day. So our last macromolecule is the nucleic acids. So this group had 5 different colors of paper clips. They had 2 different color beads, and then they had small and longer pipe cleaners, all the same color. And they made nucleotides, DNA and RNA nucleotides, and then they made DNA and RNA. So let's look at how this works. So how were our beads different? They were 2 different colors, and your beads represent 2 different sugar molecules or monosaccharides. So they had ribose and deoxyribose. So ours were clear and white. So they had this sugar on one, ribose, and they had this one, deoxyribose, the clear one, on another. So white and clear. Different, okay? Deoxyribose is missing a hydroxyl group, so it's missing an oxygen, deoxygen, so OH group is missing. And we have 5 different color paper clips. And the paper clips represent 5 different nitrogenous bases that form the nucleic acids, and adenine, guanine, cytosine, thymine, and uracil. And in these structures, do you see the differences? Well, let's look. So yeah, we see lots of differences here. In this one, we see this difference. We see this difference. This one's different. This is different. Okay, so you see all the differences. And we use the first letter of these to abbreviate A, G, C, T, and U. So we put the small piece of white pipe cleaner to represent the phosphate group. So here you see that we have a piece of pipe cleaner, white pipe cleaner attached to the white bead, and that's our phosphate group. And this one has a white pipe cleaner attached to the monosaccharide, which is the phosphate group as well. So nucleotides, the monomer of the nucleic acid, have 1, 2, or 3 phosphate groups attached to them. Nucleotides, the monomer of a nucleic acid, always has 3 components. So it has a phosphate group, a sugar, and then your base. So there's 3 things on this model. There is the phosphate group, which is the pipe cleaner. There is the sugar, which is your monomer bead. And then you have your base, which is your amino acid from up here. These are your bases, nitrogenous bases. Okay, so your nitrogenous base, adenine, guanine, and so on. That's this paperclip, 3 parts, 1, 2, 3, of a nucleotide. Let's see. So in this one, there's guanine, and so this is a deoxyribonucleotide. In this one, there's adenine. It is also a deoxyribonucleotide. This one right here is cytosine. And then you have thymine, adenine, cytosine, guanine, and uracil. Okay? And that's how they differ. Because you see that they all have a phosphate group. Looks like they all have the same phosphate group. They all have the same sugar. Well, no, they don't. This one's different. So this one, that is what makes this one not a deoxyribonucleotide. So this one has lost its OH, lost its OH, lost its OH, lost its OH, making all of those deoxyribonucleotides. This one still has an additional OH. This one does too, so that makes them just ribonucleotides. And so this one both have their OHs, so those are also ribonucleotides. So let's look at how this works. So in this, your adenosine and your thymine and your A and your T go together, and your C and your G go together. So let's look and see if this model is made correctly. This is a model of your DNA. And you can see that we have the pipe cleaner for the phosphate group, we have the bead for the sugar, and then we have the paper clip for the nitrogenous base. Now, if I say that the A is green and the T is pink, then I should have an A green with a pink T. And that's not what this one is. This one's not correct. If I have a pink right here, then what should be attached to it? A green, because the A and the T go together. Let's look at this. I have a blue and a blue. No. If blue is cytosine, then it wants to go with guanine. So I should have a blue and a yellow. And if I have a yellow, I should have a blue. So anywhere there's a green, it should be attached to a pink. And anywhere there's a blue, it should be attached to a yellow, because those are the way that the nitrogenous bases pair. The adenine always goes with the thymine, and the cytosine always goes with the guanine. So we'll work on getting this model repaired so we have it for the future. All right, let's look at this page. You were to color the ribose, the sugars dark red. So let's look at all of our sugars. Sugar, those are those monosaccharides. There they all are, your sugars. And then your deoxyribose, dark blue. Here it is without your OH group. So those are your deoxyribose sugars on this model. Your phosphate groups we circled in yellow. So here's a phosphate group. There's all your phosphate groups. And then it wanted you to do your guanine green. Now this is what I'm talking about. So if your guanine is green, then your guanine needs to go with the cytosine. So green and pink go together. Do you see how green and pink go together here? And if blue is adenine, then it has to go with the thymine, which is yellow. So blue and yellow go together. That's how our model is not correct. We need to fix this model. Okay. So then we go into RNA. So when you make a single strand, let's see where my single strand. Oh, here it is. So when I make my RNA, here it is right here, I only have 1/2 of it. I still have my phosphate group, my sugars, and my nitrogenous bases, but it's not combined with the other half. So using your RNA strand, okay, we're going to stop here. I'm not going to continue on with RNA. Just know that there's a different nitrogenous base called uracil here. And that it looks like half of a DNA. Okay, and we'll study more about this in another module. Okay, so that brings us to the end of all of our macromolecules. I do have a slide here. Let me go over here to here. And let me share that if I haven't already. How do I do this? Okay, so it says I'm sharing. I hope you can see this. I can upload it as well. But this is a review of everything we just did. So we have our proteins, we have our carbohydrates, we have our nucleic acids, and our lipids. And these are just some notes about each one that we just went over with our pipe cleaner bead paperclip activity. I'm also going to upload some homework. I'll label it homework, go through it, and answer the questions and bring it to class on Wednesday. Go ahead and bring in your study guide for Wednesday, your flashcards for Wednesday, your Bible verse for Wednesday, because we're going to finish up Module 2 on Wednesday. So again, bring the homework that I'm uploading now on macromolecules, bring your study guide, your on your own questions, your Bible verse, and your flashcards to class on Wednesday. Okay, I've got to go move my car. It was a pleasure, and I hope that y'all have a great Monday and Tuesday. See you soon.
So what